In this video, we're going to learn about the structure of different types of lipids. So the lipids we'll be exploring are triglycerides, phospholipids, sterols, and waxes. Like carbohydrates, lipids are made of carbon, hydrogen, and oxygen, but in very different ratios, and we'll look at that in the next part of our video. Lipids are insoluble in water, but they're soluble in nonpolar substances because they're hydrophobic compounds. Lipids also provide 2 times the energy per gram than carbohydrates or proteins, which is why we often suggest that you need to limit your lipid intake because there's a lot more calories in lipids or fats than carbohydrates and protein. Our first type of lipid is triglycerides. And triglycerides are also known as fats and oils, and they provide energy for us. So triglycerides are composed of glycerol, which is this molecule right here, and 3 fatty acids. And they're also combined through condensation reactions. And in this case, the bonds between the glycerol and each fatty acid is called an ester linkage. And if you've taken organic chemistry, you'll understand why it's called an ester linkage, because glycerol is an alcohol, so the hydroxyl is interacting with the carboxylic acid. And the combination of an alcohol with a carboxylic acid is an ester, and it's a triglyceride in this case. So we end up with this. Each of the alcohol groups will lose 1 hydrogen, and the hydroxide will be lost off of each fatty acid, and then you end up with 3 water molecules being produced in the process of producing that triglyceride. So 3 fatty acids are combined to 1 glycerol. The first type of triglyceride we will explore are saturated fats. And saturated fats have all single bonds between the carbon atoms in the fatty acid chains, and they're said to be saturated with hydrogen. Because there's no double bonds, every single carbon has the maximum number of hydrogens that it can hold, and that's why it's called saturated. These chains are straight in shape, and they attract one another in that molecule because they're all lined up together, and they attract each other through London forces. If you remember back to our biochemistry video, London forces is the only type of interaction in nonpolar molecules. This attraction between these chains causes the fat to be solid at room temperature. So sources of saturated fats are animal fats, so dairy and meats and all of those animal fats would be considered saturated fats. Unsaturated fats have at least 1 double bond in the fatty acid chains between the carbons. So in this case, they're not saturated with hydrogen. So if you look at this example right here, this is unsaturated. When a double bond forms, there's electrons being shared between those carbon atoms, extra pair, so now they can't bond to as many hydrogens. So this part of the molecule is not saturated with hydrogen. Each double bond causes a kink in the chain, and it causes the chain to bend, and that will decrease the London forces between the nearby chains because they're not all nicely lined up together. This reduced attraction causes the fat to be liquid at room temperature. And sources of unsaturated fat are shown here. So fish are a great source of unsaturated fat, especially cold water fish, so salmon and tuna, etc. And then also vegetable oils. Now there are a few plant-based oils that actually have some saturated fats in them, coconut oil being an example, but mostly vegetable oils have unsaturated fats. Polyunsaturated fats mean that there's more than 1 double bond in the fatty acid chain. So that's where the polyunsaturated comes from. Our third type of fat is trans fat. So these triglycerides, we tend to say that they're man-made, although they can occur slightly in small quantities in nature. And in trans fats, the unsaturated fats become partially hydrogenated, so hydrogen can be added to fill up the double bonds. And some of the double bonds in this process get rearranged, so the hydrogen atoms are now arranged across the bond. So normally, an unsaturated fat is a cis configuration. So if you think of it, cis starts with the letter C, and this almost forms a C shape. So it's a cis bond. In trans fats, the bond flips, so now we have a hydrogen going across the bond. So one hydrogen's on one side and one's on the other. The way I like to remember it is trans means across, so Trans-Canada Highway. So that's the highway that goes across Canada. Trans fats were created by lab scientists thinking that they were creating a healthy alternative to saturated fats and butter. So margarine was one of the first trans fats used. And unfortunately, we'll talk about some of those problems, but trans fats weren't the miracle food that scientists thought they were inventing. Trans fats thicken and solidify oils and increase the shelf life of packaged foods. So that's a benefit to people producing foods that they want to last a really long time. So one example of a food that has trans fats would be McDonald's French fries and a lot of packaged foods. Mind you, there's a lot less trans fats in foods now because we're becoming more and more aware of their health risks. Diets high in saturated fats seem to be linked to raising our low density lipoprotein or LDL cholesterol levels. And LDL cholesterol is considered the bad cholesterol. Unsaturated fats raise our high density lipoprotein or HDL cholesterol levels, and that's the good cholesterol levels, and it also works to lower our LDL levels. Trans fats, however, are considered doubly bad because they are shown to lower our HDL levels, which is our good cholesterol, and raise our LDL levels. So it creates a greater gap between the two levels. And for cholesterol health, we need to have high levels of HDL in our blood and lower levels of LDL to prevent heart disease. Now, there have been claims of a correlation between both trans fats and saturated fats and coronary health, but the question has been raised through a variety of studies: is saturated fats so bad for us? Because there are a lot of places in the world where diets high in saturated fats are consumed and people don't have coronary health. So is it just a correlation? Is it correlation or is it causation? So we have to ask that question. And we're now seeing a lot of links between diets high in carbohydrates being linked to coronary heart disease. And we'll take a look at that a little bit later in one of our article studies. Our next type of lipid are phospholipids, and phospholipids are essential to the structure of cell membranes and the functioning of the cell. And phospholipids trade out one of the fatty acids for a phosphate molecule. So it's glycerol plus two fatty acids, and then a phosphate group is substituted. And phospholipids have a hydrophilic head. So this part, that's the phosphate group. So phosphate is very hydrophilic, so it's soluble in water. And then it has two fatty acid tails. And those fatty acid tails are insoluble in water, so they're hydrophobic. So this molecule is very confused because part of it wants to interact with water and part of it wants to avoid water. So when you add phospholipids to water, those phospholipids form structures known as micelles or spheres. So the hydrophilic head dissolves and interacts with the water, and the hydrophobic tails all mix and mingle into the center of that structure, and they're protected. Phospholipids go on to form our cell membrane and the membranes that surround all organelles. And the cell membrane is a phospholipid bilayer. So it is two layers, bilayer, of phospholipids. So the heads are found on the outside of this bilayer, and they interact with the watery matrix that's found both inside and outside of the cell. And the tails are protected on the inside. So you can see this part's the hydrophilic part, the extracellular space, it has a lot of water, the cytoplasm has a lot of water, and then the hydrophobic tails are found in the center and they're protected against that water environment. Water can't pass through the cell membrane because of this, so we have to develop little pores that allow the water to go through. And cell membranes have to have embedded proteins and these hydrophilic pores to allow water, ions, and polar molecules through. So if you take a look here, each of these is a phospholipid, so right here. And then these are our nonpolar tails and our polar head. And then we have a bunch of proteins that embed themselves into the membrane. So this one is called a peripheral protein. So it's just on one side of the membrane. It doesn't go across the membrane. This is a channel protein, so it goes all the way through, and it would have a pore or some sort of carrier mechanism that will carry the molecule through. And those channel proteins can be passive, so they can move the molecules across without using energy, and some of them actually have to expend energy to do so. So those ones would be called ion pumps. And we'll learn about ion pumps later in this course. We can also have what are called glycoproteins, and these are cell markers. So if you remember from grade 11 biology, and we learned about antigens or markers on blood cells, that's what that's referring to. So we have markers on all of our cells to communicate with other cells and to communicate with other molecules, so we know that things belong to us. So the markers on our cells, our body recognizes as us, so our immune system won't attack them. So glycoproteins are really important to have. Also, you'll see here, cholesterol is embedded in the protein, and cholesterol's job is to change the fluidity of the membrane. So it's found in animal cell membranes. It's mostly hydrophobic, so that's why most of it is embedded into the fatty acid tail portion of the phospholipid bilayer. But then there is a part that is hydrophilic, so it extends out towards the outer part, and it decreases the fluidity of the membrane, and it changes the permeability to some hydrophilic ions, so it prevents hydrogen ions and sodium ions from traveling through. It also helps the membrane curve, which allows the formation of vesicles. And if you remember, vesicles help surround and bring things into the cell or send out materials from the cell. There are many functions of proteins that are embedded in the cell membrane. So one of them could be a hormone binding site. So an example of that would be insulin binding to a protein on the liver. So it's a receptor that allows the cell to take in glucose to create glycogen. We also have immobilized enzymes found on the epithelial villi cells. So those enzymes are proteins that perform certain functions. So as the food is moving past, those enzymes are right out there ready to interact with the particles that need digesting. Cell adhesion, so adhesion, remember, is sticking together, so it holds cells together, forming tight junctions. They also act as channels for passive transport, so both for facilitated diffusion channels, and then finally pumps for active transport. So cholesterol belongs to our next type of lipid known as steroids or sterols. So steroids are 4 fused hydrocarbon rings. So you can see them right here, 1, 2, 3, 4, and functional groups. So in this case, there's a hydroxyl. So cholesterol is an important component of the cell membrane, as we've mentioned. And then cells can also convert cholesterol into vitamin D, bile salts, hormones, and so on. So steroids are extremely important in many processes in our body. Our last type of lipid would be wax. And wax consists of long-chain fatty acids linked to alcohols or carbon rings. And these molecules are hydrophobic, and their job is to create waterproof coatings. So the cuticle found on leaves would create a barrier to keeping water from evaporating from the leaf, so as a protective layer.